Thank you, Dr. Kazrak. It fills my heart with joy unspeakable to be here. It feels to be at home. I think this is the third year in a row we are here. Wonderful. So before my talk, I'd like to give a standing ovation to Alan. What a wow. Again, great. Please. Please. We are all here for patience, right? So, let's see. Revolutionizing rare cancer drug development with novel tumor-agnostic classifiers and AI innovations. These are my disclosures. So in 2024, November, there is a revolution going on in oncology, or rather an evolution. We moved away from the light microscope, and we are currently in the era of molecular microscope. And as Alan shared in the previous talk, we are still struggling with the molecular microscope and NGS and testing. But having said that, we are moving to the next era of AI era microscope. Treatments are evolving from one-size-fits-all chemotherapy to histology-dependent targeted therapy to tissue-agnostic drug development. So what is tumor-agnostic therapy? Tumor-agnostic therapy is a different way we think about cancers. Targeting the target, looking in the target, not the tumor. Beyond the organ, beyond the histopathology, beyond the tumor. There was this Nature paper earlier this year. Forget lung, breast, or prostate cancer. Why tumor naming needs to change? The conventional way of classifying cancers according to the organ of origin is denying and delaying people access to drugs that could help them. There are hundreds and thousands of rare cancer patients who do not have access to many drugs because those drugs, although that might work for them, are just approved in lung, breast, prostate, and kidney cancers. What are tumor-agnostic therapies? It's a different way we think about cancers. We are used to traditionally thinking as breast cancer, kidney cancer, prostate cancer, lung cancer. And historically, most cancer treatments were developed to treat a cancer that has been developed in a specific organ. Look at these targets, NTRK, BRAF, and RET. These are age-agnostic and tissue-agnostic targets. And we are entering into an era of tumor-agnostic precision oncology. In the next 50 to 100 years, I envision that there's going to be hundreds and hundreds of these targets. How do we get there? If you see all these targets, RET, NTRK, or BRAF can manifest as different tumor types across different age groups. They can occur in common cancers, rare subset of common cancers, or in rare cancers like infantile fibrosarcoma, they could be pathognomonic. Infantile myofibromas, they could be pathognomonic. They are seen across the board. Today in 2024, as of June 2024, we have over 10 drugs, not one, not two, but 10 drugs approved in a tissue-agnostic manner. This is a landscape of tumor-agnostic therapies approved by the US FDA and EMA. Let's talk about that and how this is going to revolutionize rare cancer drug development. The first one was pembrolizumab. Then the next one was larotrectinib, entrectinib, pembro again, dostarlimab, dabrafenib, trametinib, selpercatinib, and the latest antibody-drug conjugate, trastuzumab deruxtecan, repotrectinib. And we have many, many studies being designed. So if you see the thing, this is my Twitter handle. Alan mentioned about social media. This is my Twitter handle, but I see also that Blue Sky is dramatically going up. So I have an account in Blue Sky, and that's my Blue Sky handle. I post both in Twitter and Blue Sky. So among all these 10 tissue-agnostic drugs, I've been involved one way or the other. So these are my personal perspectives on tumor-agnostic drug development. Every target is a pan-target. Tumors don't follow rules and are malignant snowflakes. The reality today is there are no undruggable targets, as superior chemistry has brought us therapies aimed at KRAS and P53 mutations. Every drug, every target should be considered for tissue-agnostic drug development. Why? Because patients would not have access if the drug is not approved. Once a dose is established, organ-agnostic biomarker arms should be explored in parallel to tumor-specific cohorts and studies. We need to learn from the past. Patients cannot wait for studies in every disease. Human screening studies are important. We need to learn from the past. The future of tumor-agnostic precision oncology, this is my prediction. There's going to be more tissue-agnostic targets, more drugs with tissue-agnostic activity, more disease settings where tissue-agnostic therapy might be applicable. We are entering in an era of AI. AI-based technology is going to unleash more tissue-agnostic targets. And of course, critics would say there's going to be exceptions. Of course, there are exceptions in medicine. These are resistance mechanisms, but these resistance mechanisms can be addressed with combination therapy. It's not a one-size-fits-all approach. We need to make personalized treatment decisions for every patient standing in front of us. So the new era of tissue-agnostic drug development was driven by genomics. This represented, as Dr. Kurzrak would say, a marriage of two fields, immunotherapy and genomically targeted therapy. May 23rd, 2017, right, the date is important. Why? For the first time ever in the history of oncology, a drug was approved based on a target, not based on a tumor type, right? So FDA approved pembrolizumab, an anti-PD-1 antibody for solid tumors based on this MSI high or dMMR signature. Why this was important? This was a tissue-agnostic approval. It was an approval based on genomic biomarker. Approval was based on a single-arm study, not based on a randomized study, and it was an age-agnostic and tissue-agnostic approval, both in adult and pediatrics. The next was story of picking needles in haystacks, the NTRK story. Larotrectinib was approved in 2018, entrectinib was approved in 2019, repotrectinib became the third kid on the block, the latest one. Let's come to that on June 13, 2024. Again, these three drugs, not one, two, but three drugs are available for patients with NTRK fusions. NTRKs are needles in haystacks. NTRK fusions are found in diverse cancers. Again, they are seen in common cancers like lung cancer or breast cancer, but they are pathognomonic in rare types of breast cancer, like secretory breast cancer or salivary gland cancer, which we call it as mammary analog secretory carcinoma. In pediatrics, right, they are seen in deadly diseases like pontine glioma, in multiple subtypes of sarcoma. The estimated prevalence is anywhere from 1,500 to 5,000 patients in the US alone. Globally, the burden is going to be like 20 to 30,000 patients a year. The marriage story continues. Pembrolizumab was once again approved for high tumor mutation burden cancers. Another drug, dostarlimab, was approved in August 2021 for the same indication. This shows the distribution of BRAF V600E. BRAF V600E is again an age-agnostic and tissue-agnostic target. It is seen in multiple cancer types, including colorectal cancer, non-small cell lung cancer, histiocytosis. Again, we designed one of the first ever basket studies, prospective basket studies with a single agent vemurafenib for all BRAF V600E and showed that BRAF is a tissue-agnostic target. More recently, we did the ROAR basket study, the Rare Oncology Agnostic Research Study. Again, we took 9 different rare cancers and enrolled patients who harbor a BRAF mutation, deadly cancers like anaplastic thyroid cancer. Again, this shows the response rates across many rare diseases. So this data set, based on pooled analysis of 3 different studies, led to the FDA approval of dabrafenib and trametinib across BRAF V600E mutated cancers. Look at that. Anaplastic thyroid cancer is one of the most deadly cancers known to us. 56% response rate. Biliary tract cancer, over 50% response rate. High-grade glioma, glioblastoma, 33%. Low-grade glioma, 54% response rate. Adenocarcinoma of the small intestine, another rare cancer, 54%. So a pooled analysis. At the same time, the NCI Match published their tumor-agnostic cohort showing a 37% response rate, and there was another pediatric study that showed in responses of the combination in pediatric studies. So we did a pooled analysis of this drug. We reviewed the literature, submitted all the evidence, and eventually we were able to secure age-agnostic, tissue-agnostic drug approval. Of course, there is an exception of CRC, and even in CRC, BRAF V600E is druggable with the addition of EGFR inhibitor. This drug, dabrafenib and trametinib, was originally approved in melanoma in 2013, May 29th, 2013. This was always cited as, BRAF was always cited as an exception to tissue-agnostic drug development. We were able to secure a tissue-agnostic approval on June 22nd, 2022. It took almost 10 years for a drug that's already been FDA approved to get a tissue-agnostic approval. Think about those rare cancer patients who did not have access to the drug. Again, this was done in niche boutique academic centers. How many patients can travel to niche boutique academic centers to gain access to therapy? Think about that salivary gland patient with BRAF V600E in New Mexico. Think about that brain tumor patient, that pilocytic xanthogranuloastrocytoma patient in North Dakota with a BRAF V600E alteration who does not have clinical trial access. In real world, patients have access to therapy only after regulatory approval. So it took 10 years for us to get here. Again, I would say, again, as an investigator and from the patient point of view, this is unethical. We should not be doing this. We can be faster. So the next set of targets I worked was RET. RET, we know that it is activated by two mechanisms, RET fusions and RET mutations. Again, they are seen in rare cancers like medullary thyroid cancer and rare subsets of common cancers like pancreatic cancer, lung cancer, among others. Clearly, they are tissue-agnostic targets, including the zebras, right, the rare cancers. We showed that selpercatinib is a highly potent and selective RET inhibitor, showing good brain-penetrant activity in preclinical models as well. This shows the waterfall plot. So what is the waterfall plot? The waterfall plot shows the percentage, the graphs show percentage from baseline of the tumor shrinkage. So clearly you can see that this RET inhibitor, selpercatinib, is a tissue-agnostic drug. And RET fusions are tissue-agnostic targets. Look at the length and breadth of tumors that were seen. Salivary gland cancer, carcinoid cancer, small intestine cancer, cholangiocarcinoma, sarcoma. Clearly, RET fusions are tissue-agnostic targets. We took the lessons learned from the BRAF V600E, so we were able to get an approval just over 2 years after the non-small cell and thyroid cancer approval in May 2020. In the peak of the pandemic, we were able to secure an approval in non-small cell lung cancer and thyroid cancer based on expansion cohort of the phase 1 study. And the same expansion cohort of the phase 1 study, once we reached the threshold of the number of patients, we were able to secure an FDA approval on September 21st, 2022. I'm going to come back to the date, why this date is important. Jim would be happy. So FDA approval, again, recently, it was approved in a tissue-agnostic manner in pediatric patients as well, pediatric patients who harbor RET fusions. So May 29th, 2024, this drug was approved in pediatric patients as well. But you see these waterfall plots here. There is a patient behind every waterfall plot. You see that patient with pancreatic cancer who had 100% response rate. Pancreatic cancer, as you know, it's a tough disease. Again, although we have many KRAS drugs in drug development, still it's a deadly disease. This was a CT scan of a patient with pancreatic cancer. And this is the waterfall plot of that same patient with pancreatic cancer. But if you see that, behind every waterfall plot and CT scan, there is a patient and a family behind it. Meet Alison Cuban. She was a patient with pancreatic cancer, young girl, and she received standard of care therapies, FOLFIRINOX, gemabraxane, and unfortunately, her tumors progressed, and she came to our clinic. We did sequencing, and she hit the tumor harbored the RET fusion. Internally, I was happy, but I had to maintain the composure, calmness, and I spoke to her. She's like, I'm going to start this study in a month. I said, no, you need to start it tomorrow. He's like, why do you want to wait for a month? I know I won't make it. I'm going to get married next week. I said, go ahead with your wedding. Let's start the study tomorrow. She started. The wedding took place 10 days later after she started the drug. She was wheelchair bound, full of belly fluid and ascites, and she sent videos from her wedding. She got off from the wheelchair, she danced, she threw out the wig, she started growing hair in just 10 days. 5 years out, she's written a nice book, and like a needle in a haystack. She's a keynote speaker. Again, these are the stories that keep us awake in the morning. And the book starts with her being diagnosed with cancer on the same day her boyfriend, fiance, proposed to her. And then it ends by me calling her, thanking her. When the drug was approved, I call usually my patients, all the patients, and the family members if they are not around, and thanking her. And she was, again, this was a fantastic book. Again, September 21st, 2022. And that call I made from Boston. You know why? Because September 21st, the think tank on advancing precision medicine happened in the same place. And there's Jim and Dr. Kurzrak. We'll come back to the date for next year. So these are currently the targeted therapies that are approved in multiple solid tumors. Again, these are all pan targets and pan tumors. We currently live in an era of antibody drug conjugates, and this is the first ever antibody drug conjugate that's received approval. But there are challenges in tissue-agnostic drug development. Again, it's mainly for investigators and pharma. There are many unknown unknowns. So how can we move the unknown unknowns to known knowns? We need a format. We need a guideline. Every pharma company, every investigator, every academic center needs a guideline. The world is divided into breast cancer doctors, kidney cancer doctors, prostate cancer doctors. There is not a tumor-agnostic doctor. Probably all the tumor-agnostic doctors are just in this room here. Right? So who is a tumor-agnostic doctor? Everyone is a tumor-agnostic doctor. Just for that, together with the ESMO Precision Oncology Working Group and with Dr. Kurzrak here, we put together the ESMO Tumour-Agnostic Classifier and Screener, a tool addressing tumor-agnostic potential of molecularly guided therapies. Again, this is open access. Please feel free to go read that. We provide everything in depth. This is a QR code. Just Google ESMO Tissue-Agnostic Classifier and Screener. You will get that. What does it provide? It provides definitions. What is tumor agnostic? What is tumor modulated? What is tumor restricted? Also, it has a proposed framework for tumor agnostic drug development framework. Principles for tumor agnostic drug development. Biology informed mechanistic, rationale, early regulatory advice, adequate trial design, followed by confirmatory evidence. We have detailed out each and every step in detail in this tumor agnostic drug development. We were able to, come up with a consensus with our esteemed ESMO colleagues. And this is the first part. We are going to go with another second part and third part, and it'll be coming out in the next couple of years. So again, although these are needles in haystacks, the prevalence is 1%. If you add up all these tissue-agnostic targets, it's over 20 to 25%. So we are entering into a paradigm of tumor-agnostic precision oncology where we have the right target with the right biology, with the right drug, and clear benefit, there is a drug approval. So I just spoke about genomics, but cancer is beyond that. We need a complete molecular portrait of every patient walking in the door. Again, this is a story of 6 blind scientists and the elephant. Again, gender balanced. Dr. Kurzrak wanted to make sure that it was completely gender balanced. And again, right now, all the clinical trials are, it was at the proof stage, by the way. Again, I didn't realize that. Again, I cannot see what the brain does not know, but Dr. Kurzrak reminded us that it has to be gender balanced. So right now, the clinical trials, we design our clinical trials centric. We need to move to patient-centric clinical trials. As Alan said, patients need to have a voice, and it has to be patient-centric clinical trials. We need to think about more and more direct-to-patient clinical trials. Social media, there's a huge role of social media here. How can we, fix the puzzle of rare cancer zebras, right? The jigsaw puzzle. Solving the zebra puzzle through social media. Patients in rare diseases are in diverse geographic locations across the globe and can be interconnected via social media. We need to think globally and act locally. Again, one of the major advances, regulatory advances, is the accelerated approval, right? Again, this has hit the target for precision oncology and helped patients with rare cancers. The current timeline of drug development of 10 to 15 years, again, is unethical, unsustainable, right? We need to do it in 3 years. Yes, we can do it, right? I think hopefully AI can take it there. I said earlier, every patient who walks in front of us, right, we provide shallow medicine, even today in 2024 November, because we don't have all the information about the patient. We just use like shallow evidence and treat the patients. We need deep personalized medicine. Before I conclude, I think my time is almost up. Before I conclude, this is the Edwards checker box. Can you see the difference between A and B? Maybe I should pick up a student. Are there any students here? Everyone's a student, right? So which is darker square, A or B? Anyone? A is a darker square, right? How many votes for A is a darker square? Almost universal, great. So if you see that, right, optical illusion, A and B are the same. See that, this was published by Edward Adelson in 1995. A and B are same color. This is what the eyes can see. Oncology, the entire field of oncology is dependent on pattern recognition. Again, we take a biopsy of a tumor, naked eye, we have to see that via the light microscope or even the electron microscope, still using the human eye. We need to move beyond that. Look at this. AI has analyzed 100 million tissue patches for over 100,000 diagnostic whole slide images across 20 major organ types within seconds. AI to multimodal AI has the potential to unravel many tissue agnostic targets. I think AI can put everything together, amalgamate all this data together for us. So how can this happen? Because with multimodal data fusion, multimodal interpretability and association discovery, we can come up with more and more tissue agnostic targets and repurpose more and more drugs for tissue agnostic purposes, especially in rare cancers. All right. I work with ChatGPT, I collaborate with ChatGPT version 4.0 for sonnets. Again, precision medicine and tissue agnostic rare cancer precision medicine is still unfinished. This is still not a sonnet. Sonnet is 14 lines. Next year, I'll probably finish the sonnet. All right? So this is a unified vision, the dawn of tumor agnostic precision oncology. In Boston's halls where wisdom's torch doth brightly gleam, a vision shared by Target Cancer Foundation's light, tumor agnostic drugs fulfill the dream of tailored care for rare cancers, precision noble fight. Oncologists with science keenness art chart pathways where one's shadows held their sway, uniting minds, a future they impart, where hope and healing guide the modern day. In accord, their vision leads us on to realms where suffering's veil is swiftly gone. I use ChatGPT to come up with dream, gleam, and the rhyming words, and some of the lines are my own. So again, the first author is ChatGPT and the senior author is me. Again, in summary, every target is a pan target. Every drug should be considered for tissue agnostic drug development. Why? Because patients with rare cancer are missing huge. We need to learn from the past and live in the present, but think about the future. AI. Patients cannot wait for trials in every disease. The ESMO Precision Oncology Working Group proposed a framework to foster and accelerate tumor agnostic drug development. I thank Jim, working with NCCN, working with ASCO to come up with tissue agnostic guidelines for across the board. ESMO proposes the minimum requirements to screen for tissue agnostic potential. AI to multimodal AI has potential to unravel many tissue agnostic targets. Again, before I conclude, September 2021, we got the selpercatinib approved. And last year, November 15th, 2023, again, another drug was approved. and I didn't know that. Another drug was approved, repotrectinib. And this is also a tissue-agnostic drug because the drug was eventually approved this year, again in June 2024, as a tissue-agnostic drug. Again, some random connection with numbers, but again, something Jim picks up some dates, keeping fingers crossed for today, if we can get a tissue-agnostic or a drug approved today. I think there is a drug, PDUFA date is end of this month. In fact, Jazz is here, Elena is here this morning. We convinced Jazz to, they are doing a tissue-agnostic study. It's called Discover, because they're working on a biliary tract cancer drug, and we are doing a tissue-agnostic drug across all HER2-positive cancers in the setting. So again, thank you. Thank you to all the patients, all their caregivers, to travel long distances for these clinical trials. We need to get to some point where patients have access to clinical trials where they are by their local oncologists so that they can sleep in their own beds. Thank all colleagues, co-investigators, and Dr. Kurzrock, Jim, for inviting me for this talk. Thank you so much.